Okay, everybody, welcome to lecture number 3, where we're going to talk about DNA, race, and identity. So let's get into the topic for today. So DNA technology emerged in the 1970s and has really kind of grown, especially over the past 20 years. In 2007, the New York Times published more than 800 articles containing the term DNA, just to give you a sense of how popular this idea in news media has become. Genetic research has also fueled the genealogy business, challenging older ways of knowing ourselves and our ancestry. With a simple cheek swab, my genes can be compared to populations from around the world, unlocking the mystery of my ancestry, or so they promise. Despite what family history, photographs, or archival records might show about our ancestors, genetics makes possible really bold claims about our personal and familial identity. But can your DNA really tell you where you're from? Take a couple minutes to think about how you would answer this question. Where are you from? Often when I'm asked where I'm from, I first think about where I was born, which was Pittsburgh, Pennsylvania. But I also think about where I grew up, which is in the suburbs of Washington, D.C. I often don't think about my ethnic background, but I think that's often what people are asking when they pose the question, where are you from? So this is the kind of starting point where geneticists are really trying to understand ancestry. They're trying to answer the question, where are you from? So let's talk a little bit about how geneticists start to answer that question. And we have to begin by understanding what a gene actually is. So a gene is a segment of DNA that contains the message for a protein. Genes are the building blocks of our bodies, the basic units of information that tell our bodies what to do. Genes provide the primary means by which individuals pass on biological information to their children. And differences in genes create biological variation, and they are the drivers of biological change. So you can think about genes as being the drivers of human evolution. Genes are segments of the filaments of deoxyribonucleic acid, that's a tongue twister, which is made up of molecules that reside in each of our cells in the form of these spiral-shaped strands, which are known as a double helix, right? So the double helix is what our DNA looks like and was first identified by scientists in 1953. DNA is condensed into chromosomes, which are basically supercoiled little masses of DNA in the nucleus of our cells. DNA has two main functions, replication and protein synthesis. Replication refers to the ability of DNA to copy itself. DNA copies help create new cells, which is what's needed for our bodies to heal and grow. Protein synthesis refers to the creation of protein molecules, which are made up of amino acids. The DNA is the code, or basic kind of set of instructions that tell our bodies the right sequence of amino acids needed to put together in order to create proteins. Each human genome is made up of 3 million base pairs of genes spread across 23 chromosomes. 99.9% of these genomes are similar to one another, which means that the genetic difference across the human population is 0.1%. To put it another way, you may have Senegalese ancestry on both sides of your family, going back as many generations as anybody can really remember, but you have the same genetic makeup as someone who is Scandinavian, Japanese, or Melanesian. That 0.1% difference in our DNA sequences across people is not random, but is inherited from your parents. These kind of differences are referred to as polymorphisms and are inherited in equal parts from our mother and father's DNA. Each of these different messages from our maternal and paternal lines produce the very slight variations which we see across individuals and which cause differences in physical traits, or what's called phenotype. Our current genetic diversity reflects what biological anthropologists call gene flow. Gene flow is basically the movement of genetic material within and between populations. So you can see in the diagram behind me, the little blue bird is moving across the lake into the red bird area and is mating with the red birds and creating gene flow. So the migration of individuals from one population to another can alter genetic variation in all the populations involved. Populations that are geographically distant, for example, Mexico and China, tend to have less gene flow than populations living nearby. So think about France and Germany, the United States and Mexico, things like that. Gene flow can occur through non-random mating, right? So when people choose their partner, they can either decide to stick with their immediate group or choose a mate with specific traits they are attracted to in a different group. So let's take an example of gene flow. So the Hutterites are a Protestant group in Canada, very similar to the Amish here in the United States. And they live in these kind of small, rural farming communities. Because of their cultural and religious beliefs, they seldom marry outside of their immediate group. Anthropologist Michael Park examined over 1,000 fingerprints from 2 different Hutterite communities that split from the founding group in 1958. What Park found was that these 2 splinter groups were actually more genetically similar to each other than to the founding colony because they were geographically closer to each other, and individuals between these 2 splinter groups married people within these groups. So gene flow had happened between these 2 offshoot colonies, which reduced genetic variation between the offshoots, but increased genetic variation from the original Hutterite community. Gene flow is essentially how we get variations in DNA across populations. The first serious proposal that genetic variation existed between human populations came from Richard Leviton, who's depicted here behind me, who developed this theory in 1972. Leviton analyzed differences in the appearance of certain known genes and compared these against one another. A key contributor to advancing Leviton's theory has been the Human Genome Project. So the Human Genome Project is an international scientific research project which was in operation between 1992 and 2003. The primary goals of this project was to identify all the genetic material in modern human populations. The driving force between the Human Genome Project was the goal of identifying disease genes and developing better predictors of disease and associate treatments. Sorry, I'm just looking to see what my dog is chewing on. My bad. Okay. What their research has shown was that between 83 and 97% of genetic variation is found within populations, and between 3 and 17% is found between populations. So I'll say that again. Most genetic variation is found within populations rather than between populations. Importantly, the scientists of the Human Genome Project argued that the concept of race had no genetic basis and that information from the project should be used ethically and fairly. While geneticists have identified DNA sequence differences that predict continental ancestral origins, what DNA reveals about race is far from obvious. As we'll talk about next week, in the 20th century, genetics and race were conflated through the social science of eugenics. While in academic circles, scholars argue that race can't be defined biologically, in popular culture, race is often equated with ancestral DNA markers. Within popular applications of genetic science, race has also been equated with disease, geography, and what we think of as ancestry. So let's take a quick break from the lecture and fill out the attendance quiz. So if you're watching this at home, make sure that you go onto D2L and fill out the quiz question, which is, what is the artifact for today? And the answer is Scythian hoard. So what you're looking at are solid gold artifacts that were discovered in a Scythian burial mound in southern Russia. And Scythians were basically these kind of fierce equestrian warriors who had control over the grasslands of Eurasia for about 1,000 years and were recorded in early Greek oral histories by scholars like Herodotus. All right, so make sure you fill out that D2L quiz to get credit for watching the lecture today. All right, let's get back into DNA, race, and disease. So many geneticists use self-identified racial categories published by the Federal Office of Management and Budget to discuss disease occurrences and outcomes among Americans based on race. These categories are used by the U.S. Census Bureau and are based on a mixture of skin color, geographic origin, and shared language. The OMB openly states that there are sociopolitical categories rather than biological categories, right? So Japanese is a sociopolitical category, not a biological category. But a recent study of 30 geneticists showed that scientists still use these categories as racial types in their studies. Well, some researchers argue that there is an established connection between genetic background and disease susceptibility, which is important to study. Others say that linking race to disease can result in the stigmatization of whole populations. So what actually is the connection between race DNA and disease? So relatively rare inherited diseases like sickle cell anemia or cystic fibrosis, which is caused by mutations in a single gene, are often linked to racial groups. For example, 1 in about 700 African Americans have sickle cell anemia compared to 1 in 150,000 Europeans who have it. These are both genetic disorders that are caused by recessive mutations in genes. But if we look a little bit closer, the racial basis of these disorders is actually a red herring. Sickle cell anemia is a disease that occurs when an individual inherits a specifically mutated version of the 6th amino acid in their beta globin gene from each of their parents. This mutation arose independently in different populations at least 5 times during human evolution. So we see it in Bantu, Benin, Senegal, Cameroon, and Indian haplotypes. A single mutation will provide a slight advantage to children in resisting the lethal effects of malaria, which is why scientists think that this mutation occurred in the first place. So what you're looking at in this graph behind me is an image that's comparing the prevalence of sickle cell anemia and the presence of malaria. So malaria, places in the world that have high rates of malaria are the dark green areas. So you can see that there's a lot of dark green areas in Africa, but also on the Indian subcontinent. And then you can also look at the map above where you see the prevalence or frequency of the genetic mutation that can lead to sickle cell anemia. And again, you see that it's primarily located in Africa, as well as hotspots in the Middle East and in the subcontinent of India. So this kind of single mutation would have been actually a selective advantage, which would be passed on and multiplied over time within the populations that were highly susceptible to malaria. And this mutation would have resulted in a greater likelihood that a child would inherit a mutant allele from both parents, which would cause sickle cell anemia. So a single mutation doesn't cause the disease and would have been a selective advantage. But over time, as more and more people with this single cell mutation intermarried, the likelihood that their offspring will receive a double mutation increased, leading to this mutation that actually has negative effects on health. The major source of African slaves were from the very regions where high levels of sickle cell beta globin alleles were present in the population. And this mutation was brought to the Americas through the chattel slave trade. So from a genetic perspective, then the sickle cell anemia is associated with individuals whose ancestors originated in specific malaria-prone parts of West and Central Africa and Central India, not African Americans as a racial group. For most diseases in the US that differ in incidence by race, racial differences actually also correspond to socioeconomic and cultural differences. For example, most of the differences in the incidence of asthma, hypertension, and heart disease by race can be explained by income disparities and environmental risk factors that marginalized populations face. What this tells us is that individuals with disease risks are linked less closely to one's genetic makeup than to factors like age, their biological sex, environmental and behavioral traits, linked to things like income, diet, smoking, and residence. Over the past 40 years, geneticists and anthropologists have used genetic studies of people from geographic and culturally isolated groups around the world to define origins and migration of human population. Populations are defined by these specific sequences in either mitochondrial DNA, which is your maternal DNA source, or Y chromosomes, which is your paternal DNA source. These studies have produced what's called the Recently Out of Africa model of human history. The Recently Out of Africa, or ROA model, says that all modern humans originated in the Rift Valley in northeastern Africa. Small groups then migrated at different times out of the Rift Valley over the past 100,000 years, moving through Africa as well as to Europe, the Middle East, Arabia, the Indian subcontinent, and eventually Asia and North and South America. Each of these groups of migrants carry specific genomic variants that become more frequent as the founding population expanded. So here's a question for the group. If you're live with me, we'll talk about it in class. If you're watching from home, take a minute to think about how you would answer. So does our common ancestry in Africa mean that race doesn't matter? In addition to research on continental ancestry, scholars have identified certain DNA markers that differ significantly between populations living on different continents, and these differences likely arose after migration out of Africa. For example, there's an allele called the FYO allele that's found as part of the dark gene, which is present in more than 90% of West Africans, but is found in less than 5% of Europeans or Asians. To identify these differences, geneticists gather genomic material from contemporary individuals around the globe. One influential study using this technique looked at 377 genetic segments in the DNA of over 1,000 people from 52 different geographical populations. What they found was that 95% of the genetic variation in the data across these 1,000 individuals was within the populations. Using these sorts of markers, the researchers then put these individuals into subgroups based on their distinctive DNA. The result is that genetic variation corresponds to major geographic regions of Africa, Eurasia, East Asia, Oceania, and the Americas. So does having DNA, East Asian DNA, make you Asian? What do you guys think? For the rest of the class, I want to talk a little bit about the relationship here between DNA testing and what we've been talking about so far, which is these racial and identity and ethnic categories that often map on to geographical ancestry. So genealogical DNA testing first became available on a commercial basis in 2000 and has since become increasingly popular, with companies like ancestry.com and 23andMe developing DNA kits. So there's 3 types of DNA that can be used to trace your genetic ancestry: mitochondrial DNA inherited from your mom, Y chromosome DNA, which only appear in men. So if a woman takes the test, it will only tell you about the paternal side of her family, and what's called autosomal DNA. So rather than looking at these 2 biological sex chromosomes, autosomal DNA looks at the 22 pairs of autosomal chromosomes which exist across your gene pool. So this is a type of test that 23andMe and ancestry.com use. They use an autosomal test. Remember from our lecture on Tuesday, though, that when we look at ancient DNA, we're only using mitochondrial and Y DNA, not autosomal. So once you send in your DNA to a lab, what the scientists do is called genotyping. Because 99% of our DNA is exactly the same, what genotyping does is to look at thousands of specific markers or areas of our genetic code. These markers are known to vary between individuals, and some are associated with specific traits or geographical ancestry. These markers are then compared to other sequences with a reference database. The test will give you matches with genetic cousins and admixture percentages based on the frequencies of certain variations. So ancestry informative markers, or AIM, testing is another type of analysis for understanding ancestry. AIM testing runs a statistical comparison between your genetic markers and a database of genes from populations around the world. The results of this test will tell you what percentages of supposedly pure old world identities you embody. The search for ancestry is particularly popular in the United States among African Americans, who have been denied knowledge of a large portion of their history and heritage due to their ancestors' forced migration to the Americas, followed by the undocumented genetic admixture that accompanied slavery. This search for ancestry was made popular by Harvard professor Henry Louis Gates Jr., who developed a TV show called African American Lives and Faces of America. In Faces of America, Gates Jr. tested the genomes of famous Americans and presents them with a pie chart of their ethnic makeup. For example, Mexican-American actress Eva Longoria went on the show and is told that she was 70% European, 27% Asian, and 3% African. She's then told that this percentage makes her genetically similar to Parisian-born cellist of Chinese parentage Yo-Yo Ma, who has almost the exact same makeup. So to which she responds, he's Mexican, which really captures the tension between genetic claims and personal ways of identifying. In the article that you guys read today, Barbara King argues that we shouldn't put very much stock behind ancestry testing. Do you guys agree? If you're at home, please make sure to go and write down your thoughts about this. What we'll do in the next section of class, if you're live, is a small group discussion based on the reading. So if you're at home, make sure to take some time and write down your responses to these questions. In class, we'll break into small groups, and you guys will have a chance to talk about these issues with people at your table and nearby, and to write down some of your responses to share with the group. So One reason why DNA testing isn't that reliable is the nature of genetic inheritance itself. We inherit half of our DNA from each parent, and stepping back a generation, we get a quarter of our DNA from each grandparent. Each of our siblings also inherits a quarter of each grandparent's DNA, but it is not necessarily the same quarter. So we will only have partial overlap between siblings or cousins. If we do the math working back 200 years, that means 256 different ancestors contributed DNA to each of us. Can DNA testing tell us about the geographic origins of all 256 distant ancestors? Well, most DNA companies test DNA sequences from either mitochondrial DNA or non-recombining portions of the Y chromosome. These two tests at best provide information about 2 out of these 256 ancestors. Those companies that look at markers scattered throughout the chromosome spectrum, so that's autosomal testing, right, that looks at all 23 chromosomes, can provide information about your paternal and maternal ancestry. But we don't know how many ancestry markers there are or how many should be tested. That means depending on which variants an individual has inherited from their 256 ancestors, the same DNA test could give different geographic ancestry results for siblings with the same parents. Ancestry testing also has some more technical issues. So the ability to situate individuals in the past and link them to particular places based on genes is more limited for certain people and certain regions. For example, the sampling of individuals from sub-Saharan Africa, North and South America has been really limited. So the likelihood that if you had these ancestries, it would show up in an ancestry test is small because the comparative gene pool is smaller. Determining ancestry beyond the continent level is a statistical exercise based on differences in frequency of variants at multiple independent locations on a DNA strand. The presence and absence of a specific allele at any one location on your DNA strand is usually insufficient to determine ancestry. The job of ancestry analysis is to figure out which expected pattern of variation among tested population groups best matches the set of variants present in your DNA. The reliability of the observed population data completely depends on the sample size and representativeness of the standard group analyzed for each population. This gets really tricky if an individual's ancestors come from totally different populations. The more missing different ancestral populations in an individual's family history, the less similar a person's genetic pattern will be to the standard population. Before geneticists began searching for identity, we had a well-established and complex way of knowing ourselves. This explains the skepticism that many have towards genetic revisions. But genetic knowledge need not be set above cultural and political ways of thinking about identity. Selective editing interpretation is just as prevalent in genetic analysis of identity as when using archives or photos. So genetic analysis is as much about making meaning in the present as it is about finding any truth in the past. All right, that's all I have for you guys today. I hope you enjoyed the lecture, and I'll see you next Tuesday.